John, a 55-year-old man, come to doctor visit due to the difficulty in swallowing for past several months. John has no history of the chest pain or heartburn, and he lost 5 kg weight in the last 3 months. Physical examination remain unremarkable. On endoscopy, ulcerated mass in the distal third of the esophagus was present. The biopsy of this mass and adjacent normal mucosa were performed. Analysis of this biopsy show accelerated cytosine deamination of the chromosomal DNA in both normal and malignant cell. This damage is most likely to be repaired through the which enzyme sequence. Before going to the right answer, first we have to know the detail in the DNA repair mechanism. The first thing is clear in this question that there is a problem in the DNA, the cytosine deamination. So when cytosine deamination takes place, it converts to the uracil. So the uracil is not present in DNA. Uracil is only present in RNA. So the DNA repair can classified into two types. So one is a double-stranded DNA repair one, and another is a single-strand DNA repair. So single-strand DNA repair further divided into the three type: nucleotide excision repair, base excision repair, and mismatch repair. So I will discuss the mechanism of these single-stranded repair DNA in detail later. First, we discuss about the clinical correlation that this nucleotide excision repair is defective in xeroderma pigmentosum. So this is defective in xeroderma pigmentosum. While the base excision repair is basically due to the deamination, basically the deamination reaction due to the multiple causes. It may be spontaneous or due to exogenous nitrates. So last one is the mismatch repair. So what is the mismatch repair? So mismatch repair is basically the example of the mismatch repair is the clinical correlation example in which the mismatch repair is defective is the Lynch syndrome. So all of these three things are super high yield. So as we already know that the single standard DNA repair is three type: nucleotide excision repair, base excision repair, and mismatch repair. So the first, what happened in the nucleotide excision repair? In this nucleotide excision repair, what happened that this is a thymidine dimer. So UV exposure and any other changes that lead to the pyramidal dimer, then this dimer is removed by the endonucleases enzyme. And finally, this newly replaced segment is done through the DNA polymerase and the ligase fill the gap. So the two enzymes are involved for this newly replaced segment, DNA polymerase and ligase. So this type of the nucleotide excision repair takes place in the G1. So next is the base excision repair. So base excision repair occur throughout the cell cycle. And the next important thing is that here is the deaminated base. The deaminated base, when you heard the deaminated base, so in the clinical vignette, they give this deamination of the cytosine. So deamination of cytosine lead to the formation of the uracil. So now I'm going to discuss about the cytosine uh, deamination repair. So the cytosine deamination lead to the cytosine deamination convert the cytosine into the uracil, and the uracil nitrogen base is present in the RNA. So this is a abnormal. So what happened? There is an enzyme known as a glycosylase. So this glycosylase enzyme remove this deaminated base and form the AP site and lead to the formation of the AP site. And next is the two another enzymes, AP endonuclease and AP lyase. So AP endonuclease cleave the 5 end, this end, while the this lyase cleave the 3 end. So this 3 end is cleaved by this apelase enzyme, and this 5 end is cleaved by this AP endonucleases. So the sequence of the enzymatic process is in the deamination is the first is a glycosylase enzyme, then is the AP endonuclease, and then lyase. And finally, this base excision repair is that DNA polymerase fill this gap, and after that the ligase seals this repair. So again, this is the high yield stuff that deamination of the cytosine lead to the conversion of the cytosine into the uracil. What happened? This glycosylase enzyme recognize this deaminated nitrogen base and remove this, and there is a AP site appear, and this AP site and the surrounding of this nucleotides are removed by the 5 end is through the AP endonuclease, and 3 end is the lyase. And next is the DNA polymerase. So DNA polymerase fills the gap. DNA polymerase fills the gap, and the ligase is the last enzyme that seal it. The sequence of this is first glycosylase, endonuclease, lyase, polymerase, and the last one is ligase. So this is a high yield stuff. To sequence of this, and the what happened? This deamination is basically due to the maybe it's endogenous, and maybe it due to exogenous, mainly due to the excessive nitrogen, nitrate. Sorry, so excessive nitrate. So the last one is the mismatch repair. So the mismatch repair here is the adenine pair with thiamine. Normally, this pair with thiamine. In this, the adenine does not pair with the thiamine, and here is the mismatched nucleotide is the guanine. So what happened? This mismatched segment is removed and the newly segment is added. So normal thiamine is added. So if this mismatch repair is defective, it will lead to the Lynch syndrome. Lynch syndrome. So now I'm going to discuss about the certain other nitrogen base and there is deamination products. So as we already know that cytosine deamination lead to uracil, while the adenine deamination reaction lead to hypoxanthine, and last one is a guanine. Guanine deamination lead to the xanthine. So the deamination of the nitrogen base lead to the activation of the base excision repair mechanism of the DNA. So the sequence of the base excision repair of the DNA is the first glycosylase. Glycosylase identify the deaminated nitrogen base and it is removed and lead to the formation of the AP site. Next, the endonuclease. Endonuclease cleaves the 5' Next is the ligase. It's cleave the 3' And next sequence is the polymerase. And last one is the ligase. So basically, this is the sequence that happens in the base excision repair of the DNA. So this sequence match from the option D. So the option D is the right answer of this question.